Welcome to the biology class, children. How are you all? Today we are discussing three topics. The topics are outbreeding devices, pollen pistil interaction and double fertilization. Okay, so we are starting with the topic outbreeding devices. Majority of flowering plants are hermaphrodites. Hermaphrodites means both the male and female gametes are produced within the same flower. Okay, and it increases the chance for self-pollination. So what happened when self-pollination increased? It results in breeding depressions. Okay, and it reduces the quality of offsprings and it reduces the chance for variation. To discourage self-pollination, flowers develop different types of devices to encourage cross-pollination. So today we are going to discuss about these devices, and these devices are termed as outbreeding devices. Okay. The first feature is unisexuality. So in unisexual flowers, self-pollination is not possible, but we know that plants may be monoecious or dioecious. So in that condition, bisexual flowers, anthers and stigma mature at different times, so to prevent the self-pollination. So depending on who matures first, dichogamy can be further divided into protandry and protogyny. In protandry, anthers matures earlier than the stigma of the flower, whereas in protogyny, stigma matures earlier, so they get pollinated before the anthers of the same flower develop their pollen grains. So it helps to prevent self-pollination. Then third condition is heterostyly. In some plants like oxalis, the stigma and the anthers are placed at different levels. It also helps to prevent the pollen from reaching the stigma and pollinating it. Another mechanism is pollen prepotency. Prepotency means in this mechanism, the pollen of a different flower germinate faster than the same flower, thus prevent autogamy. The next one is self-incompatibility. In this mechanism, it is a genetic mechanism, okay, and it prevents self-pollen from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in the pistil. Okay, that was about pollen outbreeding devices. Next, we are moving to a new session, that is pollen pistil interaction. We know that pollination does not guarantee the transfer of right type of pollen, right? Pollen of the wrong type, either from other species or from the same plant, if it is self-incompatible, also can be landed on the stigma, isn't it? So the pistil has the ability to recognize the pollen, whether it is of the right or wrong type. If it is the right type, the pistil accepts the pollen and promotes the post-pollination events that led to fertilization. So if the pollen is of wrong type, it rejects the pollen by two methods. The first method is preventing the pollen germination on the stigma or by preventing pollen tube growth in the style. Children, do you remember our practical pollen tube development? Okay. So if the pollen is of wrong type, it prevents the pollen tube growth in the style. Okay. Thus it retards the movement of male gamete towards the embryo sac. Clear? Then, the ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. That means a lot of interactions are happening between the pollen grain and the pistil. So, we are going to discuss about this interaction. In compatible pollination, pollen grains germinate on the stigma to produce a pollen tube through the germ pore. So we know that except at the germ pore, sporopollenin, the tough organic material is absent. You remember, children? So the contents of the pollen grain move into the pollen tube. What is the content of pollen tube? It contains the male gametophyte, right? So the pollen tube grows through the tissue of the stigma and style and reaches the ovary. So we know that there are pollen with 2-celled stage and with 3-celled stage. So this is a diagrammatic representation of a pollen with 2-celled stage. In this 2-celled stage, the generative cell again divide and form 2 male gametes. And in 3-celled stage, the pollen carries 2 male gametes from the beginning itself. Okay, so after reaching the ovary, so here the movement of pollen tubes, you can see the pollen tube enters the ovule through the micropylar end and enters into one of the synergids. So we know that inside the ovary at the micropylar end there are two synergids. And this movement is through the filiform apparatus. What is filiform apparatus? It is a chemical substance present inside the synergid. Okay, so this filiform apparatus gate the entry of pollen tube. This filiform apparatus is capable of checking the species specificity of the male gamete. Clear? That is pollen pistil interaction. So children, along with this topic pollen pistil interaction, I have to mention one more topic, that is artificial hybridization. If a breeder is interested in crossbreeding, he can produce superior varieties. Okay, for that he or she can opt artificial hybridization technique. It is one of the crop improvement programs. So in it, desired pollen grain has to be used and dusted over stigma. And this process is achieved through emasculation and bagging. This topic, emasculation and bagging, is very important the point of view of practical examination. Okay, so first we can discuss emasculation. What is emasculation? If the female plant bears bisexual flowers, removal of anthers from the flower bud before the anther dehisces by using a pair of forceps, that is emasculation. Okay, so we can use desired pollen over it, isn't it? And in diagram you can see that process, and it is followed by bagging, the next process. Emasculated flowers has to be covered with a bag of suitable size and should be made up with polythene cover or butter paper, okay, to prevent contamination of its stigma with unwanted pollen. When the bagged flower attains receptivity, desired pollen can be dusted over it. Okay, but in the case of unisexual flowers, no need of emasculation. We can do only bagging. Okay, only bagging can done. So this topic is very relevant. Okay. So today's last topic is double fertilization. Okay, double fertilization is a unique phenomenon found among angiosperms. Okay, now we can listen the chart. Double fertilization is a unique phenomenon found among angiosperms. On entering to one of the synergid, the polar tube releases two male gametes. So here filiform apparatus are present to check the specificity of male gametes. One among the male gamete combined with this egg cell. This is egg cell. So we know that egg cell is haploid in nature. This egg cell is combined with another haploid cell, that is male gamete, and produce diploid cell, that is zygote. And this process is termed as syngamy. This process is known as syngamy. Clear? The remaining one male gamete move towards the central cell and combine with the polar nuclei located in the central cell. So, the remaining male gamete combine with two polar nuclei present at the central cell and produce primary endosperm nucleus, PEN. So this process is termed as triple fusion. Triple fusion. Children, why this process termed as triple fusion? One nuclei present in the male gamete combine with two polar nuclei and produce PEN. That's why this process termed as triple fusion. Let me ask a question. What will be the ploidy of PEN? Yes, it will be triploid. And what will be the ploidy or zygote? It will be diploid. 2N and this is 3N. So two processes, both syngamy and triple fusion is happening during this process. That's why it is termed as double fertilization. Clear? This PEN after triple fusion, it become primary endosperm cell and it develops as endosperm, whereas zygote develops as embryo. So the process of double fertilization is clear, children? Children, today we discussed three topics: outbreeding devices, pollen pistil interaction, and last one was double fertilization. Don't forget to draw each and every diagram to your biology notebook. Study the textbook and notebook well and answer the questions. If any doubts are there, ask me. Okay. So we can meet in the next class tomorrow. Next class is about post-fertilization events. Okay. So till then, bye.